Good morning. Please let me know if you can't hear me or see the screen. Thanks. It is a real pleasure to be here today, and thank you for this previous presentation. That's very important work. We've been working on coronaviruses, I guess me since 1984, but we've been working on approaches to understanding the basic biology of coronaviruses, particularly their replicase, and looking at antivirals for the last, I think, probably 20 years, really, based on the basic questions that we've been asking, and really very intensely in the last 6 or 7 years. So everything I'm presenting today is really fundamentally predicated on that. I just want to mention our lab and the people that are working really very hard around the clock. I know everybody has different challenges, and ours has been sort of an overabundance of work and expectation over the last 2.5 months. So the people on the team, particularly Maria Agostini, recently graduated student, Jim Chappell, and Andre Prousers, Laura Stevens have been highly focused, but the entire lab has been as well, people who've been here previously. So I wanted to be sure I noted that. Okay, let's see if this works. Next slide. Okay, as well as our collaborators at Chapel Hill, University of North Carolina, Chapel Hill, Ralph Barrick, who I've been collaborating with for over 20 years, Amy Sims, Tim Sheehan, and Rachel Graham, and then collaborators at UAB, Gilead Sciences, our funding, and at Emory University. I'll make a brief shout out to them at the end. Our work is really based on, as I mentioned, this is sort of from a 2018 U19 grant application, but we had it in 5 years before that, and 10 years and 15 years before that, talking about the significance and the emergence of SARS and MERS, and the capacity of coronaviruses to emerge from animals to cause severe disease. And then this issue of bat SARS-like coronaviruses circulating in nature that Ralph and others had shown, Ralph Barrick and others shown the capacity to replicate in human cells, even cause infection and potentially disease in mice. And so our strong sense that this would be a problem, and the resistance to therapeutic antibodies and vaccines that have been developed against SARS, for example, or MERS. So with increasing overlap of wild animal ecologies, the potential for human emergence is high. So this was really from our significance and impact section from a long time ago. I just want to give a brief, and I know the audience is diverse, I appreciate that. I just want to give a very brief sense of sort of, this is an older phylogeny based on the coronavirus polymerase, the main replicase protein that I'll talk about today, but just really showing the diversity. And if you look in the blue boxes, these are what are called the beta coronaviruses. And you can see that they're populated by several kinds of different viruses, including the known human viruses, HKU1, OC43, and then MERS, SARS, SARS, the new SARS, SARS-CoV-2 or COVID-19, as well as the model virus that I'll talk a little bit about. And then these viruses here I want to mention, because these are really the ones that had driven a lot of our momentum toward thinking about antivirals before the pandemic, pre-pandemic, because these were the ones that I mentioned were known, had been shown by Ralph and others to be really capable of infecting human cells and potentially being what they were calling pre-pandemic viruses. And you can see they come from the same group of this called 2B beta coronaviruses. Different names are used, but basically showing you that they're really occupied by Bt viruses, the bat viruses, and that the numbers of these are increasing as well. So this concept of the need for this is something that we have been very interested in. So our need for antivirals is obviously this broad diversity of coronaviruses in bats. Bats are over 1,300 species, probably 22% of mammals are bats. And every bat species that's been looked at carefully by really important groups like EcoHealth Alliance and others have shown that almost all bats that have been looked at are shedding 1, 2, 3, or more coronaviruses asymptomatically. And so there's a vast diversity of bat viruses. And so this work around the world is exceptionally important and is really predictive in lots of ways of what we're experiencing today, although this is certainly a worst case scenario, I would say, of what we would have even thought with our sense of what could happen. So I think this need for antivirals is the diversity. Obviously, the issue of the time to develop vaccines differs from the trajectory of the epidemic, as we're all experiencing pretty dramatically. And then this concept of the potential for off the use shelf toward highly conserved functions, and this idea of being sort of future-proof and preparing, which is really what our goal has been. We didn't think it would be something that would happen within a month or two after some of the antivirals that we proposed using. So our goals for antiviral development have really been differentiated a little bit from classical drug development. We came to this as the biologist, starting my lab, this concept of that we weren't really interested in working with any compound from a preclinical development that wasn't broadly active against all diverse coronaviruses, every coronavirus we could test. and that we wanted things to have a high barrier to resistance. We wanted things that would be robust and last long with limited genetic path toward resistance and a high fitness cost, meaning that any mutations that were introduced would lead to a loss of ability to replicate and or loss of virulence, so that those would not be competitive with the parent viruses. Extended therapeutic window, as this is another discussion, it's interesting to talk to people about this now, because all these things that we have been proposing are things that people are experiencing. So when does a drug work? Does Remdesivir work early in infection? Does it work late in infection? Does it work when you have severe disease? Does it work prophylactically? So it's really not about just if a drug works. That's unfortunately kind of a winner take all. Is it fail or does it succeed? And that really misses the nuances, the important nuances of when do you use a compound, when is it best used, and how do you combine drugs to make them work better. So this idea of having additional goals of decreasing transmission and potentially having opportunities for different modes of administration depending on severity of disease. These were the things that really have driven us. I'm an pediatric infectious disease specialist as well. So seeing kids with illness, late illness in the ICU, when we were treating them for bad viral infections like adenovirus, and seeing that we could really knock down viral replication, but we couldn't impact disease, was something that really impacted me in terms of the thoughts as a biologist of working with the virus. So what are countermeasures and how can they work? Well, obviously direct acting antivirals. So that's what I'm really described today. The idea of something that directly acts against a viral function, and that viral function ideally is something that's not shared by the host, so that you don't have off-target effects. So something you're really trying to block the virus replication cycle. And those would be something, of course, that could be used for treatment. They could be used for prophylaxis. They could be used to potentially prevent transmission, right? So there's lots of ways to prevent progression of disease as well. Monoclonal antibodies, you'll be hearing more and more about. That is, of course, taking B cells from infected or vaccinated humans, isolating their antibody repertoire, identifying by genetic reconstitution those antibody genes, synthesizing the antibodies directly and using those to passively immunize. That has, I think, very, very important potential mechanisms for example, prophylaxing, preventing spread within a population, trying to limit the scope of an infection, protecting vulnerable populations. So I think these are really, really critically important. Host-directed therapies, as people know now with the expansion of the trial that includes Remdesivir, to look at those that might be targeting late host immune responses. And so this idea that we proposed, which is the idea of direct-acting antivirals to knock down virus replication, but also approaches that would extend the therapeutic window so that you could limit the immunopathologic damage that's associated with SARS-CoV-2. And of course, vaccines with an exclamation mark. I would like to think that we would have the same kind of forward thinking, that this is not just an opportunity to target COVID-19, but this is an opportunity to look at ways that we could potentially universally identify either mechanisms for virus attenuation or strategies which could be implemented and potentially prepared ahead of time for every bat coronavirus that we identify. So I won't go into specific vaccines or approaches because that's not the goal for today, but I do think it's really critical to think of it in these terms, not just for now, but for the next one that comes, because as we know, 1,000-year flood doesn't mean it'll happen every 1,000 years. It means you have a 1 in 1,000 chance of it happening every year. And I would not have predicted that we would have had this within this time frame. I would have not predicted that we would have had it within the time frame of my career or my life. But here we are within weeks or months of us really thinking about and preparing these antiviral strategies. So I'd like to spend a little time. I hope to insult the experts in virology and with simplifying this, but to help everybody understand the areas and the targets that have really been aiming for. So I do want to go through just a slight bit of the biology, and this is, I hope, something that will help you understand as people discuss what's going on, where we're targeting. The viruses, coronaviruses, are RNA viruses, meaning they contain an RNA genome. There's no DNA in their life cycle. They replicate in the host cell cytoplasm, or we call them obligate intracellular parasites or pathogens, because they absolutely require the internalization and the use of a host cell. They cannot replicate on surfaces by themselves. They cannot survive independently. And so they're replicating 24 hours a day, 7 days a week, 365 days a year somewhere. That's just what they do. And so as with every virus that we know of. So this is critical for how we think about targeting them. Their entry into the host cell, of course, depends on the virus somehow fusing with the host cell membrane. And as you can see here, the virus binds, in this case, to ACE2, the receptor. Entry of different coronaviruses can be by at least two different mechanisms. One is a receptor-mediated endocytosis, where it's taken up into the host cell after binding. Modifications occur in the host cell in the intracellular endosomal environment, which allow for a fusion protein to fuse the virus particle, releasing the RNA. Another mechanism potentially is direct entry of the virus by direct fusion, and viruses can switch back and forth. There are some versions of coronaviruses that can use both mechanisms, and so they could escape mechanisms that are targeting the endosome. Potential targets or potential drug activities would be things like endosomal proteases like camostat mesylate or other inhibitors of TMPRSS2. Theoretically, hydroxychloroquine would work at this stage by alkalinizing the endosome and preventing virus fusion. I'm not going to spend any more time on those today. Once the virus enters the cell, the host genome, the RNA genome, enters into the host cell cytoplasm. This is, of course, a lovely schematic, but of course, a simplification of a model. And showing that the RNA genome is, the first thing that happens is it's translated into proteins. And those proteins are very, very large polyproteins that then are processed by viral proteases. Those proteins then go on to do two things, multiple things actually. One is to modify the host membranes to create factories. So they really modify the membranes of the host cell. They create factories that the virus uses for replication. The proteins also assemble to replicate and copy the genetic material, to copy the RNA genome. Those RNA genomes are copied along with other small RNA molecules, which I won't spend more time on today. And that these are used for making viral proteins. The genomes are assembled on the air-Golgi intermediate compartment and then likely secreted in large secretosomes, allowing for virus to exit the cell without really lysing the cell or causing direct damage. So, but the point of all this is that every stage of this potentially is vulnerable, as with every virus. So there's nothing unique about that with coronaviruses, but there are unique aspects that may be coronavirus specific and or may be generalized to other viruses that could be targeted. So targeting entry, targeting the assembly of the viral replicase protein, targeting its ability to modify the host membranes, targeting virus replication, assembly, and or release of the virus. These are all potential areas where we could target. So what we're particularly interested in, and I have been for many, many years, is viral replication. So what happens once the virus gets into the cell, it begins making its proteins and begins to copy the RNA genome. Are there mechanisms we could use for doing this? And the enzyme that we've been particularly focused on over the last 10 years is the RNA-dependent RNA polymerase. So that is the enzyme that is used to copy the RNA genome into RNA. It's made from the virus, our genome coming in. It's not a host enzyme. And so, and host cells do not have a highly conserved or frequent RNA-dependent RNA polymerases. The other thing is these are generally highly conserved across viruses, but they're really pretty remarkably conserved across the coronavirus. So they represent really a conserved target for interference. This is just showing a very simplified, and it doesn't include the new SARS-CoV-2 structures that are beautifully being coming out of multiple laboratories now. But what it does show is that if you just sort of, I won't go into details, but if you look at sort of red, blue, and yellow, what you can see is that you have these general structures that are conserved in the polymerases. They're called the right hand structure, or a thumb, palm, and fingers domains in the red, yellow, and blue that are the pathways through which RNA passes and to be copied. But so these are targeted because they are conserved. They're critical, essential, absolutely, for virus survival and replication. And of course, then they're targeted by what are called nucleoside and base analogs. So these are analogs of normal nucleosides that would be incorporated into the growing RNA chain. And so multiple different ones that you may know of, ribavirin, AZT, sofosbuvir for hepatitis C, acyclovir, which is I'll call it the mother of all nucleoside analogs and still really effective compound against the herpes viruses, favipiravir for influenza, and many, many others. So we, of course, had always been interested in this with coronaviruses, but the experience with SARS and MERS had been that these really didn't work. Ribavirin, other compounds just didn't work. people said they did in anecdotal cases, but in our laboratory, they had never worked in our hands. So let me show you what I mean by this. When we tested this is using our model virus, mouse hepatitis virus, but we saw the same experience with SARS. What is shown on the left here is a log reduction in virus titer, and on the x-axis is showing increasing concentrations of compound, and these are very high concentrations of ribavirin or of 5-fluorouracil. And what the theoretical curve should look, slope should look like, obviously a very idealized theoretical, is that as you increase dose, you decrease virus replication if you add it. And theoretically, you would get log reductions. So, you know, a 10-fold, 100,000, a 10,000, 100,000, up to a million-fold reduction in virus replication. And what you can see from ribavirin and 5-fluorouracil is that you see a lovely flat line that the virus says, I don't care about this compound at all, and essentially are completely resistant to it. So this was profoundly interesting to us, and we've been aware of this for a long time. You can see as long back as published by Clint Smith from my lab in 2013. And we wanted to know the answer of why. And so to make a really fun story for me, very short, I was at a meeting and starting to discuss our data and the opportunity for essentially looking at this. But why was I interested in this compound? Well, it turns out that others had predicted, and then we confirmed in our laboratory that coronaviruses encode the only known proofreading exonuclease for an RNA virus. They encode it in addition to the viral polymerase. So the polymerase is the synthetic function, and no other RNA virus encodes a proofreading exonuclease. So we demonstrated that in activation, that this enzyme is responsible for removing misincorporated nucleotides, very high fidelity replication, and that inactivation of that enzyme leads to increased mutations, a mutator phenotype, decreased fitness, and attenuation. So we then wanted to test it to see what its effect was on nucleoside analogs. And in fact, what we saw was that when we inactivated the exonuclease, if this was the curve I showed you before, the flat lines, now when we inactivate that enzyme in a mutant virus, what we see is profound sensitivity to these compounds, suggesting that this enzyme was in fact responsible for sort of native resistance to nucleoside analogs. So based on this was my questions of was it possible to find those that could inhibit the coronaviruses in the setting of its proofreading exonuclease. I like to call it its superpower because it's the only RNA virus that possesses this. And so we, through connections through Craig Cameron and others, and my initial starting interactions with Gilead in 2012, actually, and our first test in 2013, we began working, and this is just to show you that what we're working with Remdesivir is not based on something magically showing up. It was based on really our proposal to test this against coronaviruses, and then the development of this really over a 6-year period with support from the NIH and with multiple collaborators at UNC, Gilead, and UAB. Our first experience that drove us this direction was, remember the flat line? Well, this is a wild type mouse hepatitis virus. This is wild type, and our first experiments by Brett Case, 2013, showing that in fact this was not a flat line, but we saw a lot of activity. And then when we inactivated the exonuclease, that we saw even more activity, suggesting that this drug was really active against the wild type virus and able to overcome this particular function. We then went on. This was a parent drug. It's called GS441524, and it is actually the active compound in cells that is converted. Remdesivir is converted to. So this was done blindly. We didn't know what this compound was at the time, and then we were given Remdesivir as the prodrug from Gilead. And what we discovered was that it was 100 times at least more active than the parent compound, because of its ability to get into cells and modify, and up to a million-fold reduction at less than 1 micromolar. So for those of you not doing this, I didn't know because I didn't do drug development. This is just an exceptionally active, highly active compound, a million-fold reduction at less than 1 micromolar. It's not 50% reduction, it's a million-fold reduction. So this is a lot of activity with a very, very low effective concentration, 50%. So this was the parent compound, and we then began in 2014 to work with this. I'm not going to go into the chemistry, but just to show you that that the parent compound is a modified adenosine, and then that GS5734 or Remdesivir is a phosphoramidate prodrug that allows for better cell penetration and more rapid metabolism and phosphorylation to the nucleoside triphosphate form. How does it work? Well, there's some lovely work coming out now from Matthias Goeth's lab and others that directly are looking at SARS-CoV-2 and really are showing that this is what's called a non-obligate chain terminator. And I'm only saying this because you'll hear this from other compounds that are being looked at. And it's interesting. It's called non-obligate because it looks like a normal nucleotide. It goes into the growing RNA chain, and then its chemistry allows for the addition of 1 to 2 or 3 more nucleotides after that, and then it terminates. So there's something about the relationship of this to the polymerase itself that results in essentially a chain termination. So this is very effective, as you could imagine. It's not a mutagen, it doesn't induce multiple changes, but every RNA strand that gets a molecule of this is essentially going to terminate. And that's a very effective strategy against the viruses themselves. So we then had this high level of activity, many different tests to test that, testing the pharmacology. But we also then wanted to answer our next question is what happens with resistance with this virus? If it's something that happens in a single round of replication, if the virus can get by it that quickly, then what's the point of using the compound? So the question is, does it select for resistance, and what's the consequence of resistance? Well, we had, I told Gilead, after doing this and working on this for a long time, we were able to tell our collaborators and tell Gilead, we have really 2 pieces of really good news for you. One is that it's really difficult for this virus to select for resistance, very difficult. We had to coddle it and push it along and help it out, just give it a little drug at a time, take the drug away, and for 23 passages. And then what's the consequence of resistance? Well, the consequence was that if resistance occurred, it looked like the virus was weaker. And I'll show you what I mean. When we passaged the virus, what we found over a long period of time, what we found was that we could get some resistance, not complete resistance, which would have been a flat line, just some resistance, which could be overcome by increasing concentrations. But nevertheless, we could see some resistance to the virus, to the drug. But this was in vitro after 23 passages and careful. And what we then did is sequence the virus, and remarkably, we really only found 2 relevant mutations in the whole 30,000 nucleotide genome, and those were both in the viral polymerase. And we wanted to know what the impact of those was. So what I won't show you is that we took those and tested them independently and together in another genetic background, in an isogenic background of a new virus, and found that those 2 mutations were capable of mediating the resistance that we identified. The next question was, what's the impact on the virus? Well, it looks like, summary, that resistance mutations are less fit. So what did we do? We took a lot of the mutant, like 9 parts mutant and 1 part wild type virus, and put them together in the same culture. And after 1 round of replication, we got out 9 parts of mutant and 1 part of wild type virus. But then, as you see, if we took those viruses and we passed them from cell to cell to cell passage culture over time, after 4 passages, the wild type virus really won. And by multiple other passages, the mutant virus goes away completely, suggesting this virus just can't compete with its parent. In work done in the Barrick lab by Tim Sheehan and Amy Sims and others, is that this virus also was less virulent in mice. So when you gave the virus at the mice a high dose of wild type virus, you saw this weight loss and a very slow recovery. When you used the mutant virus, you saw less weight loss and a more rapid recovery. And in fact, this virus really looks like it's attenuated and less able to cause disease. So
Mark,
these were all,
just a 2-minute mark.
Okay, sure. So just to summarize a couple other things. The next thing we tested was that Remdesivir is what is it like against multiple different coronaviruses. And so this was done in the Barrick lab, where they tested it against the human respiratory coronavirus, a MERS-like bat virus, a SARS-like bat virus, and then these 2 pre-pandemic viruses, and showing that it had really the same efficacy in terms of knocking down every coronavirus that was tested. So its overall impact on virus and disease in preclinical studies was that in the murine model of SARS that was done in the Barrick lab, that IV administration before or early after infection decreased virus titer, decreased weight loss, decreased lung pathology, improved lung function. And later in infection, it always decreased virus titer dramatically, but it didn't improve lung disease or mortality. Now, this was a mouse model with a very rapid onset of disease, and so they're not humans. So this is, and then more recently, there have been a study in bioRxiv. I haven't checked if it's been updated yet and published, but decreased virus replication in the lung of rhesus macaques, but not in the upper respiratory tract, interestingly, and decreased lung virus titer and disease in macaques. And as many of you may have heard, that this is showing early preliminary results suggesting that it's efficacious in reducing the time to recovery and in reducing mortality, which is very encouraging for us because we expected that if it was used in the sickest patients that you might not see a difference if you just tested it in the most ill patients. So in summary, it inhibits multiple divergent coronaviruses. It has a high barrier to resistance. It's useful for prophylaxis and treatment in mouse models and hopefully in humans. We now have a preprint out showing it's active against SARS-CoV-2 in vitro in multiple cell types and in mice, and then that there is potential reduction of virus and disease in macaques. I don't have time to talk about an oral compound that we're working with now called EIDD 1931-2801, which is orally available. It functions as a mutagen, and it is the green stars indicate the areas where it's met milestones as well. And we have ongoing data looking at this in animal models as well. And it's an exciting opportunity to potentially have another antiviral that will go in this direction. So just to finish, this is just basically the idea that we would love to begin to look at additional direct-acting antivirals, particularly in combination of direct-acting antivirals, monoclonal antibodies, and immunomodulators, as well as opportunities to look at those that could be used in different mechanisms like oral use for prophylaxis and decreasing transmission. And with that, I will be happy to stop and answer any questions. Thank you very much.
Thank you, Mark. That was terrific. I mean, one question is that I would like to before opening it up. So the studies on remdesivir have shown that there's clearly a reduction of recovery time, but not necessarily of mortality. So could you clarify a little bit about, and then maybe with the sort of, I think there was a potential, oral or actually an inhalation version of remdesivir that was being prepared. And that may allow administration at earlier time points where, in fact, it could be more efficacious. I think one of the limitations has been that the patients receiving it have been, in fact, the ones that were already quite ill.
Yeah, well, this is when I was allowed to be. I'm not involved in the studies at all, any of the clinical trials, but I was involved in early discussions about designing of the trials. And this is really a critical question, because my essential answer was, if you're just going to use it in the people who are laid on ventilators or on ECMO, then just don't do the trial, because it will look like it fails. We know, and I think the animal models were very important, because they did show that it always knocks down virus replication, but it can't cure immunopathology, right? So if you had, I think equivalent to a fire, if you have a fire extinguisher and you spray it on the coals and you fall on the coals, you might burn yourself, but not badly. If you fall in the fire and your arm gets burned, you can spray the fire extinguisher on it, but your arm is still injured. And I know it's a trivial explanation, but I think I'm actually, I can't see any of the data. In fact, I'm hopeful and optimistic that there was this marker at all of some potential improvement, not significant in mortality and improvement in time to recovery, because that took all comers. It took moderate disease and severe disease. And so that signal was identified, to me, was exciting. It may be important to parse that data so that as the data comes out, that the moderately severe disease, I would like to predict that we will see much more of an impact on progression of disease and recovery in those who are moderately ill. And so that early use, again, would be really important. But I am also hopeful that maybe we'll see that actually can impact people even later in disease and allow them to have recovery when they may be progressing to more severe disease. So I actually was, I was remarkably pleased that there was any signal that was available, because of probably most people who were treated were severely ill.
Thank you, Mark. See, there's a question by Neville Klein.
Yeah, hi. Thank you very much for your talk. Can you give a potential explanation for the discrepancy between the lung involvement and the upper tract and the effect of remdesivir, as well as seen, I think, in some preliminary studies from Oxford with their virus vaccine?
It's a fascinating issue, right? I mean, number one, this is true of going to be true of any of these compounds. This virus is testing every system, right? And so the entry of the drug, its ability to be metabolized, is really a critical question, right, into the active form. So I can just tell you, looking at SARS-CoV-2 in vero cells versus CALU cells, which are human respiratory cells. Viros are African green monkey kidney cells. The Remdesivir is much less active in vero cells, and a lot of people are publishing that, which is not unfortunate. It's just wrong to say that it's not active, because in CALU cells it's profoundly active, okay, against SARS-CoV-2. In vero cells, not nearly as active. In fact, the parent prodrug form, GS441524, is much more active in vero cells than it is in CALU cells. So I think this may have something to do with the ability of the compound to penetrate in different areas. It might also have to do with degrees of replication. It might also have to do with, I guess, potentially access of the drug to particular compartments. I wouldn't think that would be different for respiratory epithelium than in the lung, but we don't know for sure. So I think it's going to be really important to look at that, but it really is an interesting question that's raised by the macaque studies and potentially others.
Thank you. Jingyu Zhu?
Hi, great talk. So there's been multiple reports indicating that remdesivir is a analog. So the polymerase activity was reported to be much better than the actual ATP.
Yeah.
And is that because this coronavirus polymer has some specific structure features in compared to, what's the impact to the host of the RNA polymerase if it's better substitute than ATP?
So there doesn't, if that's a question about toxicity, it appears to really be competitive for this RNA-dependent RNA polymerase. And as you know from our proofreading studies, I was shocked by this. For us, from a biological perspective, this may have identified a type of compounds or a group that are capable of actually bypassing, avoiding, or getting in so well or competing at such high levels with ATP for the RNA-dependent RNA polymerase of coronaviruses that, in fact, the proofreading function is unable to act efficiently on it before additional nucleotides are added and the chain terminates. So I don't have a structural atomic explanation for it. I think there's some that are coming out, and I need to read those more carefully too. But it's a really exciting observation that it's very favorable over ATP and may be incorporated so quickly that the proofreading function can't deal with it.
Thank you.
We have Rahul Rabadan.
Hi, very nice talk. I would like to ask you if you can comment on some of the side effects that were observed in the clinical studies, and if you observe the same thing in animal models, and if there is any expectations about
review what you're specifically talking about, because I just have seen the preliminary results as well.
I see.
But I think in the early studies with hepatitis, there was, or in HCV, I think there was some hepatic and renal toxicity associated with that in the animal models that was reversible. And so I haven't reviewed the other stuff. Do you want to comment on that, or what was seen?
Well, no, I mean, it's basically from the papers and the press and the things that I've been reading, and I couldn't find anything specific about.
So far, it's been remarkably well tolerated, and there haven't been any dramatic or serious adverse events associated with it that would have led to its withdrawal. So and in the studies that were done in animals prior to that, which I wasn't privy to all the details, I know that their data was that the changes were reversible changes. The concerns were its use. Obviously, if you're using it for 6 weeks or 8 weeks or 12 weeks in something like hepatitis C, you might be very concerned about that. But its use really acutely in human respiratory infection, if the reversible changes, is something that was what people were actually hopeful about. They were limited.
Thanks.
So unfortunately, we're running out of time. Maybe just one last question, patience. Abu Aghi.
Thank you very much for taking my question. Amazing presentation. I'm so happy. Please, I have a question, and I think it's silly, but I want to ask. Considering all the side effect that comes with Remdesivir, do you think or do you recommend that healthy carriers of this COVID-19 virus can be given this medication prophylactically? Would you recommend that?
Well, number one, it's currently an IV drug, so I think that probably healthy outpatients who are not progressing in disease would probably not be eligible for this. I could imagine you could potentially use it as an infusion if someone was an outpatient and they didn't want to be in the hospital and they might be progressing. I think the reversibility of side effects is important, and I differentiate side effects versus significant toxicities that are long-lasting. You know, if we're treating someone with adenovirus, pneumonia, and we use Cidofovir, we're going to get renal damage with a single dose. And the question is sort of what are you going to die from? And I think that's kind of where we're at with Remdesivir right now. It is IV administered. If they're likely working on, you know, inhaled versions and oral versions would be my guess right now. And you can imagine that if you could give high local concentrations to the upper respiratory tract with an inhaled version of this, that might really be very efficacious, might have much more limited any questions about toxicity or absorption, as well as an oral version, which might be you know, more bioavailable and modified in the liver to its active form. So I think that there's, what this says is that if this is promising, and if the data once it's released supports what's been seen or even enhances that, then I think we're in a position to really think about how you begin to modify and look at new versions of the drug to enhance its activity in its local places of replication.
Andy, sorry, I missed your raised hands. If you want to ask a quick question, I apologize.
Yeah, Mark, that was great. Thank you very much. Is it known why bats do not get sick from coronavirus? And is there any therapeutic insight that can be gained from that?
You know, they are marvelous animals, and their biology is as complex as multiple divergent species like cats and dogs. You know, they're very different species, and we have so little knowledge of them. My trivial answer is that the selection against virulence is so high, if they fall out of a hibernaculum or they die and fall into this pile of guano and are digested, that they've co-evolved over millions of years. And I can only draw an analogy. It's sort of like avian influenzas, right? Where most of them really are not serious diseases in birds, but that they represent sort of an ongoing mechanism for survival of the virus, and it's a high level of adaptation. I think we need to discover what that is. It may help us to understand what areas we could potentially target therapeutically or prophylactically to prevent these kinds of infections. So, sorry, I don't have a better answer for you.
Thank you.